Welcome to Study Loop.
We are being asked about a scenario in which you come up with an effective treatment by getting a virus to interact with the P53 gene. So before we look at our scenario, real quickly, let's look at what the P53 does. The P53 gene is a tumor suppressor, okay? And like all genes, the P53 gene encodes, which is up here, encodes for some sort of protein. So that protein undergoes transcription and that gene undergoes transcription and translation in order to produce the P53 protein. The P53 protein is itself a transcription factor, and it is a transcription factor for the P21 gene. I know it's a lot of acronyms, but just follow along, okay? So P53 is a transcription factor for the P21 gene, so it promotes the transcription of P21. And that results in the production of P21. The protein P21. P21 stops the activity of CDKs, which are cyclin-dependent kinases. And cyclin-dependent kinases promote division. Okay, so let's now kind of write it in shorthand. Okay, I needed a different color pen. So P53 promotes production of P21, which stops CDKs from acting. That should be a D, which promote division. Okay, so if you increase P53, let me draw a better arrow there. Should be an up arrow. Okay, if you increase P53, you increase P21, which means that you lower the activity of CDKs and you prevent cell division. So that's how P53 is a tumor suppressor gene, because if it can detect that there's something severely wrong with a cell, its production is increased, and its production results in increased production of P21. Increased production of P21 blocks the activity of CDKs. Let me not make that a plus sign, just meant to connect these two lines. So P21 blocks the activity of CDKs, and by blocking the activity of CDKs, you ultimately block division. So if you're creating a virus that is interacting with P53, the virus needs to be inside the cell, because all of this is happening inside the cell. And that's a key thing to remember when we're answering this question. Actually, there are two things we need to remember here. One is that the virus is interacting with the P53, right? Because we are told in the question that this virus is active only when P53 is mutated. If P53 is wild type, the virus will not replicate. So the virus and the P53 are somehow interacting. And the virus can detect whether the P53 is wild type or not. And two is that we are inside the cell. So now let's look at our answer choices. Choice A, the modified virus only infects and enters cancer cells. It wouldn't know to only enter cancer cells. It wouldn't know that. It would enter both cells the same, because P53 is not expressed on the outside of the cell, so it wouldn't know which cell had the defective P53 or not. Choice B, the modified virus replicates in normal and cancer cells. Well, then if that was the case, then it wouldn't differentiate between the two and it would kill all cells. And we are told in the question that that's not what it does. So choice B is incorrect. Choice C, the modified virus only infects and enters normal cells. Well, first of all, uh, again, with the entry part, the P53 is not on the outside, so it wouldn't know to only enter normal cells. But then again, that's also self-defeating, because if you're only infecting normal cells, you're killing normal cells, but you're leaving the cancer cells alone. Choice D is that the modified virus replicates only in cancer cells, and that is the only rational choice here, is because it needs to be inside the cancer cell, and it needs to replicate only inside cancer cells where the P53 gene is mutated. So choice D is the correct